Hi, my name is Aina.
Hi, I'm Ellie.
Hi, my name is Ariana.
We are the Evergreen Team.
Have you heard of ZOPHOBAS morio, or as known as superworm? It has light brown skin and lives from 6 months to a year. Do you know that besides feeding on organic food, ZOPHOBAS morio also has a bacterial enzyme in their gut that degrades polystyrene foam. Our research centers around ZOPHOBAS morio.
Polystyrene-based pollutants pose substantial threats to human health and the environment. It breaks into little pieces easily. Animals eating these pieces can die due to toxins and blockage of their stomach. Chemicals that leak into food, polystyrene containers, can affect human hormones and contribute to various health problems. The ability of superworms to degrade polystyrene foam can harness it, break it down, hence being an approach to sustain the environment. The main objective of our research project is to study the behavior of ZOPHOBAS morio in different circumstances.
First, we split the beakers into 2 parts, containing soil and not containing soil. Then, we label the beakers A, B, C, and D and place 10 worms in each beaker. We also added polystyrene foam. We positioned beaker B and beaker D in a dark cabinet and beaker A and beaker C under a direct sunlight. We recorded the observation and collected the data. After 61 days of observation, beaker A did not contain soil and was exposed to sunlight. We found that the superworms in beaker A digested the PS foam quickly due exposure to sunlight. The superworms were alive but weak due to a dry environment and lack of water. There is an increase in the size of worm. They were less active in the final observation.
Beaker C.
We contained soil and exposed to sunlight. We found that the superworms digested the PS foam slowly because it hid within the soil. The superworms could survive solely on PS foam. The length of the superworms were unchanged. They were less active in the final observation.
Beaker B did not contain soil and was kept in the dark. We found that the superworms in Beaker D digested the PS foam quickly. Some of the superworms died due to malnutrition, but the rest survived. There was an increase in the length of the superworms. They were less active in the final observation. Beaker D. Beaker D contained soil and was kept in the dark. We found that the superworms in Beaker D digested the PS foam slowly because it hid within the soil. Some of the superworms died due to malnutrition and the rest were weak. The length of the superworms increased. They were less active in the final observation.
In our research, we have found that different environments and settings do have effects on the behavior of superworms. The superworms could survive solely on PS foam, although it is an unhealthy diet.
As for innovation, you only need to set up container and 100 of superworms, or more than that is also okay, and put it in condition when there's no light and no soil. This method is actually versatile, stable for school labs, household, factories, and wastewater sites. It is cost-effective and flexible. When we feed the superworms polystyrene foam in a controlled environment, it facilitating rapid digestion without harm. This symbiotic process benefits both parties. The worm gains protein from PS foam, and we safely dispose of the foam, which takes about 500 years to decompose.
The guts of superworm are able to decompose plastic waste into smaller molecules, components. Of course, you won't easily solve plastic problem, but it offers a beacon of hope for a more sustainable future. This is an ingenious way of dealing with human-made problems, and we can learn from ZOPHOBAS morio and harness their potential.
We plan to study the long-term effects of polystyrene foam on the growth and life cycle of superworms as an added improvement to our project missions in the future.